Somewhere inside a laboratory in Cincinnati, a piece of human tissue just did something that should have taken a month. It did it in 14 days. No scalpel touched it. No donor gave it up. No family waited by a phone for a transplant call that might never come. It grew on its own, inside a plastic tray built by scientists who were trying to solve one of the oldest problems in medicine. The human body does not make spare parts. And for 70 years, that single fact has cost more lives than almost anything else in modern healthcare, until now. Because in May of 2026, a team at Cincinnati Children's Hospital published something that quietly rewrote the rules of regenerative medicine. They didn't just grow human tissue. They found a way to grow it twice as fast, 10 times larger. And with something no one expected, its own nerve cells, firing, sensing, behaving like the real thing. This is the story of how we got here, where it's about to take us. To understand why this matters, you have to understand the size of the problem it's trying to solve. Right now, in the United States alone, over 100,000 people are on an organ transplant waiting list. 17 of them will die today. Not from the disease that put them on the list, from the wait itself. The list doesn't move fast enough, because human generosity, as remarkable as it is, has a ceiling. There are only so many donors. There is only so much time. No matter how skilled a surgeon becomes, they cannot operate on an organ that doesn't exist. For decades, scientists have chased an almost science fiction idea. What if we didn't need a donor at all? What if we could grow the organ ourselves from the patient's own cells in a lab on demand? The idea sounds simple. The biology is anything but. To understand how strange and difficult this problem really is, rewind further. The first successful human organ transplant took place in 1954. A kidney passed between identical twins, a surgery so radical at the time that many doctors believed it was reckless, even unethical. It worked, and it worked precisely because the donor and recipient shared identical genetics, meaning the recipient's immune system never recognized the new kidney as foreign. That single successful operation opened a door. But most transplants aren't between twins. Most are between strangers. And the moment you introduce a stranger's tissue into someone else's body, you introduce the immune system's oldest, most stubborn instinct: destroy what does not belong. For decades, the entire field of transplant medicine has been a quiet, unglamorous war against rejection, immunosuppressant, drugs, tissue matching, blood typing. 100 small innovations designed to convince a patient's body not to attack the very thing keeping them alive. It has worked well enough to save millions of lives, but it has never solved the fundamental scarcity at the center of the problem. You cannot manufacture generosity. You cannot schedule tragedy. And so, no matter how sophisticated transplant surgery becomes, it will always be limited by how many organs the world's grief can supply in a given year. This is the wall that an entirely different generation of scientists set out to tear down. Not surgeons anymore, cell biologists, not operating rooms, laboratories. Their question wasn't, how do we transplant an organ more successfully? Their question was, what if we never needed to transplant a stranger's organ at all? The answer began, as so many answers in modern biology do, with stem cells, the body's rawest form of potential. A stem cell is, in a sense, an unwritten sentence. Give it the right chemical signals, the right neighbors, the right physical environment, and it can become almost any kind of cell in the human body. A neuron, a muscle fiber, a section of intestinal lining, a beating heart cell. In the early 2000s, scientists discovered that ordinary adult cells, skin cells, blood cells, could even be chemically reprogrammed backward, coaxed into a stem cell-like state called an induced pluripotent stem cell. It meant, in theory, that anyone's own cells could become the starting material for their own replacement tissue. No donor required, no rejection risk, because the tissue would be genetically identical to the patient it was built for. It was one of the most electrifying ideas in the history of medicine. And for nearly 2 decades, it ran directly into a wall of its own, getting those cells to organize themselves into something resembling a real, functional organ turned out to be brutally, stubbornly difficult. Left alone in a petri dish, stem cells don't build a heart or a liver or a section of intestine. They build fragments, disorganized clumps. Tiny structures scientists came to call organoids miniature. simplified echoes of real organs, useful for studying disease in a dish, but far too small, far too incomplete, and far too slow-growing to ever be transplanted into a human being. And the reasons for that were not simple engineering problems. They were some of the deepest unsolved questions in developmental biology. A section of human intestine, for example, isn't just a tube. It's a coordinated society of dozens of cell types, muscle cells that contract in rhythmic waves, immune cells standing guard against invaders, blood vessels threading through every layer like a plumbing system, and critically, nerve cells. It's an entire nervous system, in fact, so extensive and so independent that scientists have nicknamed it the second brain. Without those nerves, the intestine cannot do the one thing it exists to do: move food, sense damage, coordinate digestion. For years, when scientists grew organoids, they hit the same wall over and over again. Tissue would grow, but it would be small, fragile, simplified. It would lack the nerves, would lack the scale, and most frustrating of all, it would take for weeks or longer just to reach a usable size. An agonizing timeline when a patient's damaged organ can't wait a month for its replacement to mature. Then came the question that changed everything. What if the problem wasn't the biology? What if the problem was the container? Researchers Michael Helmroth and his colleagues at Cincinnati Children's asked exactly that, and their answer was almost deceptively simple. Instead of letting stem cells float freely in a dish, hoping they'd organize themselves into something organ-like, the team built a physical mold, a confinement system. Tiny 3D scaffolding trays engineered to force clusters of stem cell-derived spheroids into fusing together in a specific, deliberate shape, guiding chaos into architecture. They called it the confined culture system. And what happened inside those trays defied what the field thought was possible. Within 14 days, half the time of the previous method, the tissue had organized itself into centimeter-scale tubular structures, roughly 10 times larger than anything earlier protocols could produce. And they weren't just bigger, they were more complete, encoded within that structure, without being explicitly engineered cell by cell. The tissue built its own functioning nerve network. Read that again. The scientists didn't hand-place the nerves. They gave the cells a shape to grow into, and the cells did what human biology has done for millions of years. They organized themselves into a working system. It's guided only by physical structure and their own internal programming. Helmroth, a surgeon who has spent more than a decade trying to move these organoids from the lab bench toward the operating table, described the moment with a scientist's caution rather than a headline writer's excitement. He was careful to note that we are not yet growing entire organs in a tank. What the team has produced, he explained, is significant tissue that can be matched directly to an individual patient. And that, once transplanted, may continue growing and integrating as part of the person's own body, restoring function rather than simply patching a wound. That distinction matters more than it might seem. This isn't science fiction. It's something more remarkable. Precisely because it's real, cautious, peer-reviewed, published in one of the most rigorous journals in the field, Nature Biomedical Engineering. But here's where the story widens. Because Cincinnati wasn't working in isolation. Around the world, other teams have been chasing different pieces of the same impossible puzzle, and when you put their discoveries side by side, a much bigger picture starts to emerge. A picture that looks less like one laboratory's success and more like an entire field converging from different directions on the same impossible destination. At UC San Francisco and Cedars-Sinai, a separate team led by Wendell Lim and Ofer Klein has been tackling a different piece of the mystery. Not how to grow organ tissue faster, but how to tell stem cells what to become in the first place. Nature already knows how to do this. A developing embryo, a small cluster of organizer cells sends chemical signals that instruct nearby stem cells to form a heart, a limb, a brain. The problem is, those natural organizer cells only know how to build one specific structure. They can't be redirected, can't be reprogrammed to build something else on command. So the scientists built synthetic ones. Engineered cells designed to cluster around stem cells and broadcast growth signals of the researchers, own choosing signals timed, positioned, and shaped by human design rather than left to nature's single fixed blueprint. In their experiments, these synthetic organizers coaxed stem cells into forming rudimentary organ-like structures, including one that began contracting rhythmically on its own, with a hollow chamber resembling a beating heart's ventricle, a heart beginning to beat, built not from a donor, but from instructions. Imagine that image for a moment, not a heart cut from a chest and rushed across a city in a cooler, racing against a 4-hour clock before it becomes unusable. Assembling itself inside a dish because a scientist told a handful of engineered cells exactly what signals to send and in what order, and the stem cells around them simply obeyed the architecture of life itself. Meanwhile, on another front entirely, researchers have been trying to solve perhaps the cruelest limitation of lab-grown tissue: blood supply. An organ without blood vessels is a city without plumbing. Nutrients can't get in, waste can't get out. Deep inside any organoid, cells begin to starve and die the moment the structure grows too large for oxygen to diffuse through on its own. For years, this capped how big and how useful lab-grown tissue could ever become. You could grow something the size of a grain of rice. You could not grow something the size of a fist, because the center would die before the edges finished forming. In early 2026, scientists reported a breakthrough vascularized brain organoid, a lab-grown structure equipped, for the first time, with its own internal blood vessel network. Not full consciousness. Not a functioning brain in any meaningful sense, but a critical piece of infrastructure that had been missing. The biological plumbing needed for larger, more complex tissue to survive and grow beyond what was previously possible. Researchers were careful to be precise about what this meant and what it did not mean. These structures don't think. They aren't conscious. What they are is a tool, a better, more complete model of how the most complex organ in the human body assembles and sustains itself, and a step toward organoids large enough and alive enough at their core to matter clinically rather than only academically. 3 separate breakthroughs, 3 separate labs, 3 separate pieces of the same enormous puzzle. How fast can we grow it? How do we tell it what to become? And how do we keep it alive once it's large enough to matter? And there's a 4th thread running through this story, one that sounds like it belongs in a stranger kind of fiction. In Texas, researchers at UT Southwestern have been experimenting with growing human organs not in trays at all, but inside animals. The idea is called chimerism, introducing human stem cells into an early animal embryo and allowing the human cells to grow alongside the host's own developing organs. In theory, if you could coax the human cells to build one specific organ, you could grow a fully vascularized, fully sized human organ inside a living creature, one whose biology already knows how to keep an organ alive. The obstacle has always been competition. Animal cells outcompete human cells inside these embryos, crowding them out before they can build anything meaningful. It's a strange kind of biological rivalry, 2 different species cells, side by side in the same tiny embryo, quietly fighting for territory and resources. And for decades, the human cells almost always lost. But in late 2025, scientists discovered that switching off a single gene in the host cells dramatically weakened that competitive advantage, allowing human cells to integrate far more successfully than ever before. It's a small, surgical change with an enormous implication. It edges the decades-old dream of growing full-sized human organs inside another species closer to something achievable, rather than theoretical. And this isn't the first time human tissue has been coaxed to grow inside another animal. In 2023, a separate research team reported something that would have sounded like fantasy a generation ago: early-stage human kidneys, grown inside developing pig embryos, the first time a human organ had ever been produced inside another species at all. The kidneys were never meant for transplant, but their existence proved the underlying idea works. And once an idea has been proven to work once, in science, it rarely stays contained to a single lab for long. Step back now and look at all 4 threads together: faster growth, programmable identity, working blood vessels, species incubation. Now, none of these breakthroughs on their own solves the organ shortage, but each one removes a wall that used to be considered permanent. And history has shown, again and again, that when enough walls fall in the same decade, the field doesn't just improve, it transforms. There's a 5th thread, quieter than the others but no less important, and it has nothing to do with biology at all. It's automation. Back in 2018, researchers at the University of Washington built a robotic system capable of automating the painstaking, repetitive work of coaxing stem cells into organoids, work that had previously required a skilled technician's hands, hour after hour for every single batch. The robot didn't discover a new biological principle. It didn't make the tissue smarter or faster to grow. What it did was remove the bottleneck of human labor from a process that, if it's ever going to reach millions of patients, cannot depend on a handful of expert hands and a handful of elite laboratories. Automation is the unglamorous cousin of these breakthroughs, but it may be just as essential. Discovery that can only be replicated by 3 labs on Earth is a scientific triumph. A discovery that can be replicated by 300 hospitals is a medical revolution. It's worth pausing here on what this technology is not, because the line between verified science and speculation matters enormously, and this is a place where imagination tends to run ahead of the evidence. We are not yet able to grow a complete transplant-ready human heart, liver, or kidney from scratch in a lab. The organoids being produced today are matched sections of tissue, not whole replacement organs sitting in a tank waiting to be shipped to a hospital. Helmroth himself has said as much, plainly and on the record. What we have is something narrower and in some ways more powerful: the ability to grow patient-matched tissue fast enough, complete enough, to begin repairing organs that would otherwise fail. That's not a small thing. For a patient with a damaged section of intestine from short bowel syndrome, from Crohn's disease, from a birth defect that leaves a newborn without enough functional gut to survive, new tissue that arrives in 2 weeks instead of 4, that carries its own nerves that can be grown from the patient's own cells and therefore carries it dramatically. Lower risk of immune rejection is not a future promise. It's a foundation being poured right now for a bridge that engineers are already beginning to build across it. And once you can build one organ system this way, the temptation and the ambition is to ask what else. If confinement geometry can coax intestinal tissue to organize into a working structure in half the time, could the same principle apply to liver tissue, to pancreatic tissue, the kind that fails in type 1 diabetes, to the insulin-producing cells whose loss defines an entire category of chronic disease? The scientists involved are careful, appropriately, not to promise timelines, but the logic of the discovery. That physical structure alone can guide stem cells toward complexity. Researchers didn't have to engineer cell by cell, is not organ-specific. It's a principle, and principles tend to spread. Consider for a moment what it might mean for a child born with a birth defect so severe that a large portion of their intestines simply never formed. Today, these children face a lifetime tethered to intravenous nutrition, feeding tubes, repeated hospitalizations, a body that can never fully process food on its own. A therapy built on this science wouldn't just extend their life, it could hand them back something as basic and as profound as the ability to eat a meal without machinery, keeping them alive. That is what these 14 days inside a plastic tray are ultimately in service of, not an abstraction, not a headline, but a specific child, in a specific hospital bed, whose future just quietly got a little brighter.